In the evolutionary arms race between pathogens and hosts, we almost always focus on genetics. We talk about receptor binding, immune evasion, and spike proteins. But there's a physical parameter that overshadows these biological mechanisms, one that acts as the ultimate gatekeeper for whether a virus can jump from an animal to a human. That parameter is temperature. Today on Fun Facts Online, we are looking at the physics of pandemics, specifically the concept of viral thermostability, and how the thermal dynamics of a bat's flight or a human's fever determine the fate of a spillover event. This is not just about biology. This is about the thermodynamics of contagion. To understand how a virus jumps from a bat or a bird into a human, we first have to understand the physical paradox of being a virus. A virus particle, or virion, is essentially a package of genetic material wrapped in protein. To be successful, this package has to solve what we call the Goldilocks dilemma. It requires two contradictory states of being. First, it needs high stability. It has to protect its fragile DNA, or RNA, from UV radiation, dehydration, and enzymes while it floats through the air or sits on a surface. If it is too fragile, it dies before it finds a host. However, the second requirement is instability. Once that virus enters a host cell, it must be able to fall apart. It needs to disassemble, or uncoat, to release its genetic payload. This state is called metastability. Think of a virus like a hand grenade with the pin pulled. It is stable enough to be thrown, but it is under immense internal tension, waiting for a specific trigger to explode. That trigger is often a change in pH or, crucially, a change in temperature. This is where zoonotic spillover becomes a biophysical gauntlet. Biological organisms operate within strict thermal windows. Humans generally maintain a core body temperature of 37 degrees Celsius. When we get infected, we mount a fever response, pushing that temperature to 39 or 41 degrees. We often think of fever as just a symptom, but biologically it is a thermal filter. It is designed to cook out pathogens that cannot replicate efficiently at high heat. For a virus to jump into humans, it must survive this thermal filter. The problem arises when the animal reservoir, the animal the virus comes from, has a body temperature that is already higher than a human fever. This brings us to the reservoirs that keep scientists up at night: birds and bats. Birds naturally run hot, with baseline temperatures of 40 to 42 degrees Celsius. If a virus evolves to replicate efficiently in a bird's gut at 42 degrees, a human fever of 39 degrees is not a defense mechanism. It is a comfortable, cool environment. But the most fascinating case study is the bat. Bats are the reservoirs for Ebola, Marburg, Nipah, Hendra, and SARS-CoV-2. Why bats? The answer lies in the flight is fever hypothesis. Bats are the only mammals capable of true powered flight. This is metabolically expensive. When a bat flies, its metabolic rate increases 16-fold, generating massive amounts of waste heat. A flying bat's core temperature spikes to 40 or even 42 degrees Celsius. In most mammals, sustaining that temperature would cause heat stroke and tissue damage, but bats have evolved to tolerate it. For a virus living inside a bat, this creates a rigorous evolutionary training ground. Every single night when the bat flies, the virus is subjected to fever-level temperatures. Any viral variant that cannot withstand 40 degrees Celsius is denatured and eliminated. The only viruses that survive are the ones with highly stable capsid proteins and polymerases capable of functioning at extreme heat. When this heat-hardened virus spills over into a human, our primary defense mechanism is rendered useless. The virus enters a human. The human body raises its temperature to 39 degrees to fight it off, but the virus is already adapted to survive 42 degrees. To the bat virus, the human body feels like a relief. This allows the virus to replicate unchecked, which often triggers the human immune system to panic and launch a cytokine storm, causing severe disease. We see this dynamic play out with filoviruses like Ebola and Marburg. These viruses are housed in fruit bats and can replicate efficiently even during the simulated heat of flight. Interestingly, Ebola presents a nuance in thermostability. While it is robust inside the hot environment of a host, it is environmentally fragile. It relies on direct contact with body fluids because once it dries out on a surface, it degrades. However, inside the body, supported by host chaperones, it is a tank. This contrasts with non-enveloped viruses like poliovirus or adenoviruses. These are the tanks of the viral world. They consist of a protein shell that is tightly packed to protect the genome. Thermostable capsids in these viruses often utilize hydrophobic interactions in their core. Unlike hydrogen bonds, which can break easily with heat, hydrophobic cores are less sensitive to thermal disruption. Some viruses, like the bacteriophage P74-26, even use lasso-like loops that physically tether protein subunits together. As the temperature rises, these bonds actually hold tighter, preventing the capsid from exploding under pressure. We can actually see these mechanics at the atomic level. Take poliovirus. It is an enteric virus, meaning it lives in the gut. It has to survive warm, acidic environments. Its capsid is stabilized by a lipid pocket factor, essentially a fatty acid, that binds inside the protein shell, locking it rigid. It is only when the virus binds to a cell receptor that this pocket factor is squeezed out, allowing the virus to become flexible enough to release its RNA. This balance between rigid stability and functional flexibility is the key to infectivity. We saw a terrifying example of this evolution in real time with SARS-CoV-2. Early in the pandemic, the dominant strain acquired a mutation called D614G. Structural biology revealed that the original Wuhan strain had a spike protein that was prone to falling apart even at cool temperatures. It was too unstable. The D614G mutation added a hydrogen bond that acted as a molecular latch. It prevented the spike from shedding prematurely. This made the virus more stable across a wider thermal range and, consequently, much more infectious. But thermostability is not just about the virus itself. It is about the environment it travels through. Zoonotic spillover often involves a transition through the environment, water, soil, or surfaces. Here, we find a surprising ally for viruses: bacteria. Research into enteric viruses has shown that they can bind to bacterial cell wall components, specifically lipopolysaccharides, or LPS, found on gram-negative bacteria. Binding to bacteria essentially armors the virus. It stabilizes the viral structure, allowing it to withstand temperatures in composting manure or warm wastewater that would otherwise destroy it. This means the microbiome of the reservoir animal plays a role in spillover. If a bat or pig sheds a virus coated in bacterial LPS, that virus persists longer in the environment, widening the window of opportunity for a human to encounter it. This brings us to the context of a changing world. As the planet warms, the thermal dynamics of spillover are shifting. We are seeing this clearly with vector-borne diseases carried by mosquitoes and ticks. Arthropods are ectotherms. Their body temperature is the environmental temperature. Viral replication inside a mosquito is temperature dependent. Warmer ambient temperatures generally speed up the extrinsic incubation period, the time it takes for a mosquito to become infectious after biting a host. A warmer world means mosquitoes become infectious faster. Furthermore, climate change is forcing species to migrate. We are seeing ecological compression, where species are moving to cooler altitudes or latitudes to escape the heat. Highland rodents are encountering lowland bats, mixing diverse thermal physiologies and creating new opportunities for viruses to jump hosts and trial their thermostability adaptations on new immune systems. So where do we go from here? Understanding viral thermostability offers us a new tool for pandemic prediction. Currently, we categorize viruses by their genome sequence. In the future, we may need to categorize them by their thermal profiles. Technologies like differential scanning fluorimetry can measure exactly at what temperature a viral capsid falls apart. If we find a novel virus in a bat that remains stable at 45 degrees Celsius, but retains high infectivity at 37 degrees, that virus should be flagged as a high-risk pathogen immediately. It possesses the hardware to bypass the human fever response. We are also applying these principles to save lives. The same engineering used by viruses is being used to design better vaccines. One of the biggest challenges in global health is the cold chain, the need to keep vaccines refrigerated. By studying how extremophile viruses stabilize their capsids, using disulfide bridges or cavity-filling mutations, scientists are engineering thermostabilized vaccines that can be transported in tropical climates without refrigeration. We are designing synthetic protein nanoparticles that mimic viral geometry but are hyperstable, creating robust platforms for immunization. The story of zoonotic spillover is often told as a biological accident, a random encounter in a forest or a market. But at the molecular level, it is a story of physics. It is the story of protein shells evolved in the metabolic furnace of bat flight, surviving the chill of the open air, and enduring the fever of a human host. Viral thermostability is a double-edged sword. It is the armor that allows pathogens to persist, but it is also a specific physical property that we can measure, predict, and potentially counter. As the interfaces between human and animal civilizations blur, understanding the thermodynamics of contagion is no longer just an academic interest. It is a necessity for our survival.